What is the immune system? What are the main parts or components, divisions of the immune system? And how does it actually work to keep us immune or free from sickness? Welcome to this short video on the overview of the immune system. This particular lecture will cover four main parts. Firstly, what is the immune system? What are its main components? So these will be cells, molecules, tissues. How is it divided? How can you categorize it? And then finally, how does this all work together to ultimately provide immunity, which is essentially being free of sickness or illness to keep us alive? Every day we are being bombarded and invaded by certain microorganisms and toxins and chemicals that will be harmful for us. So we need to have a system in place that essentially protects us from these harmful invaders. And this is what basically the immune system is. It is a system that can identify harmful chemicals or organisms. It can respond to it. It will eliminate, and it also has memory against it for the future. So this is basically what the immune system is. It's a system to protect us from becoming sick, ill, and essentially being free from infection, which is what immunity is. The components of the immune system can be broken into three components. We have cells, we have tissues, and we have molecules that help to achieve immunity. When we look at the immune system, it's broken down into two main parts. It's broken into the innate and the adaptive. The innate system is suggesting that it's always there. So regardless of what the toxin might be, what the chemical might be, but also what the microorganism might be, it's going to react in the same way. So this is sometimes referred to be as non-specific. It's also in the way that it responds, it's very quick. So it's very fast moving, a fast response. So this could be in seconds to minutes to hours. So it's very quick in its response to the particular invasion that it has. Now there's different categories or different subsections of the innate system, which I'll go to in a second. But the other thing that you need to be aware of, there's no memory. Okay, so if you keep getting infected, let's say by a type of bacteria, it's not going to respond specific to that bacteria. Even though it's seen it before, it's just going to be the same regardless. So the response here is, I need to stress, is it's non-specific. So regardless of what the problem is, it's going to be the same. When you compare it over to here, this is specific, okay? So if you are invaded by a bacteria, it will respond specifically to that bacteria or that virus or that fungi or any kind of toxin or chemical that's come in. It will come in and the immune system will react specifically to it. Now it does have memory, okay? So if you do have a invader that you've already fought off previously, it knows, it has the equipment, it has the ability to respond to it much quicker. Now it does, unlike this particular, the innate system, it needs to be primed for it to respond. So it takes some time. So if a bacteria comes in, it needs to first find it. It needs to create an arsenal to respond to it. And so this is going to take hours, days, weeks. So it's going to be very slow. Okay. So in comparison to this, the adaptive system is much slower. But the advantage here is we have memory and it is specific. Now what we're going to do is we're going to focus on each one specifically, and we'll see how they are further subdivided, and then in there you'll see the components for each one. So let's start with the innate immune system. Alright, so now we move specifically to the innate immune system, or the innate division of the immune system. Remember, this is the non-specific arm of the immune system. Now, just before I move on, the one thing I forgot to mention in the introduction is that the immune system not only is important for eliminating, identifying and getting rid of, say, microorganisms and potentially toxic chemicals, but it's also has the ability to kill off cancer cells or any cells that are essentially dysfunctional. So it has that ability to also do internally your own cells that are potentially problematic. Now, with the innate immune system, non-specific, it will react the same regardless of the type of microorganism. Let's just, for an example, we'll focus on a bacteria. So the innate immune system has kind of two lines to it. The first line and the second line. So this is essentially preventing the bacteria to invade and get inside you, okay? The two parts to it are skin and the mucous membrane. So if you essentially look at your whole body, if a bacteria tries or wants to get into you, it has to first overcome either one of these two. The majority of you on the outside is skin, okay? So skin is its own system, but it's made up of a couple of things that help prevent microorganisms getting in. Firstly, we have the actual anatomical barrier, which is the keratin. So what it really is at the bottom layer of the epidermis is you have the live cells, and they produce new cells every day, and as they move up towards the outside, they die and they become keratin, which is protein. So you just have these high packed dense layers of protein, keratin, which is just on top of each other, which makes it really difficult for organisms to try and get through. So it's that tight packing which helps prevent organisms getting into inside your live cells. But also it's going to prevent acids and bases and also chemicals from the bacteria eroding holes and getting inside. So it has a very strong protective value. Also, your skin releases certain chemicals like oils, sweat, antimicrobial chemicals to stop a lot of bacteria growing on your skin. There's also hair in strategic places like your ears and your nose that also helps prevent certain possible invasion from organisms. So we have the skin. Okay, the majority of your body is skin, so that's how that works. Moving to the mucous membrane, this is basically anywhere else where there isn't skin. And so this would be in your mouth, in your nose, in your rectum, vagina, urethra. Okay, so these are the other areas where microorganisms can get in. You can breathe it in, you can ingest it, it can go through your nose, or it could go up vagina or up into the urethra or the rectum. So to protect you from that, we have mucous membranes. Mucous membranes are essentially cells that produce certain chemicals that help stop the invasion of these organisms, in this case the bacteria. So you have mucus, it's a very thick substance which bacteria get trapped in, and then your immune system your immune cells can come along and kill it. We also have cilia, which are little hairs in your respiratory tract, which in conjunction with the mucus, the bacteria or the organisms get stuck, and the cilia will push the mucus with the bacteria up into your throat, which you generally then will swallow. That will then send it down to your stomach, and in your stomach you have gastric acid. So the majority of microorganisms would be killed off in the stomach acid in your stomach. Also, when you ingest food which would have bacteria in it, the stomach acid would kill off the vast majority of those. In the vagina, we have a lower pH, so this is through lactate or lactic acid, and that prevents a lot of bacteria growth in that region. Also around, say, your eyes and your mouth, you have saliva and tears. They are a watery substance with chemicals in it, such as lysozyme, which are enzymes, and also antimicrobial chemicals, also preventing the possibility of bacteria growth, etc. And then finally, urine. Although this is a waste product of the kidneys, it does have a lower pH, so again making it a bit harder for microorganisms to grow in bladder and urethra, and also the force of the urine being flushed out through your urethra make it a lot harder for bacteria, etc., to grow in. So these are all the first line. So they're always there, happening all the time to prevent invasion. However, if there was an invasion that was to occur, then the second line will take over. Remember, this is still part of the innate immune system. So within the second line of the immune system, we have cells, we have the physiology, which is processes that can be turned on to help stop the infection, and we have certain chemical release. Starting with cells, these are essentially the white blood cells that are produced from bone marrow, except for lymphocytes. Lymphocytes are part of the adaptive immune system. So all the rest of the white blood cells are important for the second line of the innate. The first thing, we have a group of cells, which I'll go through now, which are called granulocytes. These are made up of neutrophils, eosinophils, basophils, and mast cells. Their granulocytes means they are filled with granules. There's certain things that are inside their cytoplasm which they release to either notify the body that there's a problem or bring more cells into it. A common one would be histamine. Starting with the first cell, neutrophil. This is the most abundant white blood cell, and these are usually the first cell, the first white blood cell that comes to an area of either injury or infection. So they're very abundant, and they're the first on the scene to notify more cells to come, but also they're very good at a process called phagocytosis, which is eating up cells. Okay, so they do a lot of that. They're very good for that. So that's neutrophils. The next one are eosinophils. They're named because they are stained with an acid stain called eosin. Now, they are most important for parasites. So certain parasitic infections in the world, like schistosomatosis, where you'd find that in South America, Asia, and Africa, eosinophils would go to the point of the parasite. They can't, even though they are a phagocyte, they can't actually eat the parasite because it's too big, but they attach themselves to it, produce a whole lot of chemicals like enzymes, like reactive oxygen species, and that will help destruct the parasite. So they're very good in parasitic infections. Basophils, also a white blood cell, also a granulocyte, seems to be strongly involved with allergies. So certain allergic reactions, these cells will be more dominant, and they'll work with mast cells, particularly with some antibodies, to cause reactions to try and get rid of the allergic or the allergen that is causing the problem. They also have the ability to bring in the eosinophils at a later date, and this is where you see the recruitment of eosinophils in things like allergic asthma. Finally, in the granulocytes, we have the mast cells, which is really important. These cells are filled with histamine, and so they're going to notify the body to create inflammation. These are located in very strategic locations, like skin, mucous membrane, or under the skin, under the mucous membrane, in blood vessels. And when there is a breach of the first line, these mast cells will release histamines, which are going to cause inflammation. Now, I'll skip these guys for a second, and we'll go into the physiology. So inflammation, I've done a video on it, acute inflammation. So if you haven't watched that, please go and look at that video. But inflammation is essentially a physiological response. Again, it's non-specific to any kind of cell injury. So what basically happens when there are cell injuries or infected cells, chemicals are released. Most commonly, mast cells will release histamines, and then through a whole cascade of processes, we get more blood to the area, more exudate to the area, and more chemicals in the area, which are going to make the area inflamed, red, hot, painful, but also bring more red blood cells into it. So that's the inflammation. The other physiological response is fever. So when you are infected or have a very high inflammatory response, there are certain chemicals that are released, like cytokines and interleukins, and they also go into the blood, but they go in the blood up to the hypothalamus and tell the hypothalamus to become hotter, and that heat seems to be a physiological, an innate response to help get rid of certain infections, particular bacteria and viral infections. So that is a physiological response. Finally, some chemicals that are released in the second line are interferons and complement proteins. Interferons can be released from cells that have been infected by viruses. So it seems that when the virus comes in, hijacks your cell and tells your cell to copy itself, this cell, as it's doing so, it will release some interferons which will go to its neighbors and kind of notify the neighbors that we are infected with the virus. Please be aware of this. Don't allow viruses to come and hijack you and essentially take over your machinery and start copying. So these are special viral proteins that are released by infected cells from viruses that kind of prevent further cells being infected by viruses. Okay, but again, it's non-specific because it's not specific to that type of virus. It's just viruses in general. Complement proteins, these are a whole collective of proteins that are in your blood all the time, but they're inactive. Now, when there is an inflammatory response or an infection response, these proteins become activated in your blood, seem to go to the site of injury or infection, and all congregate together as a big group. And when they're activated as a group in a cascade, they seem to be able to put holes in either bacteria or cells that are injured or infected. So they can essentially protect you in that way through breaking holes in cells or bacteria that are present. So that's the chemicals. But also with the chemicals, just be mindful that there are a lot of chemicals mediators in inflammation, like cytokines, like prostaglandins, like histamine, that could also fit here, but these are unique types of chemicals. Finally, we go back to these few, which are dendritic cells, monocytes, and monocytes are found in the blood, which are also a type of white blood cell, but when they become activated in tissue, they become macrophages. Macrophages are very important with the neutrophils for phagocytosis, but the reason why I'm pointing these out, dendritic cells, monocytes, macrophages, what they do different to the other white blood cells is they antigen present. So when they do their phagocytosis, so they chew up, break down microorganisms like viruses or bacteria, they ingest them, break them down, but the proteins on these bacteria or viruses or microorganisms, they then present it on the outside of their membrane, which allow then the adaptive system to come in. So now the adaptive arm of the immune system will come in to the antigen presenting cells. So let's say you were infected by a bacteria. The bacteria was then chewed up, swallowed by macrophages. The macrophages break it up, but then present parts of the bacteria on its outside of the skin, outside of the cell, should I say, and then some cells from the adaptive system, most notably T cells, will come along, and now you will activate the adaptive system. So this is where the two merge together and have some crossover, which is I'm about to go to the adaptive system. But before I do, the final cell just to be aware of are natural killer cells. So these are cells that essentially just go around the body, and if they sense that either there's a cell that is infected or a cell that is dysfunctional, they will kill it or essentially tell it to die, which is apoptosis. The way that they can do that is it seems that infected cells or dysfunctional cells lack certain identification markers on the outside called MHC or major histocompatibility complex. They lack that, so these natural killer cells attach, tell them through a chemical process that you need to kill yourself, go through apoptosis, and then that happens. So that's the innate immune system. All the different parts, the two lines and the different components, which you can kind of see the components here. So we've already done one division. Now we go to the second arm of the immune system, the adaptive immune system. As we saw, this is the specific arm. There's two subsections of this. There's the humoral and then there's the cell mediated. The humoral arm, which means in fluid, was named because antibodies float around within the blood or the fluid of the body, and antibodies are produced from B cells. So both of these cells that are going to be involved in the adaptive system are lymphocytes. So this is the other two types of cells in the white blood cells. We did the whole group of granulocytes and so forth. But this is these groups are the lymphocytes. So for the humoral, we're talking about B cells. The reason why they're called B is because they mature in the bone marrow. Now the B cells are the ones that produced antibodies. But at the point before they've been activated or primed by the specific type of microorganism, in this case a bacteria, they are what we call naive. So I will leave that for a second and we come across to the cell-mediated arm. The cell-mediated arm, unlike working with antibodies, these work specifically with cells. Now these cells are the T lymphocytes or the T cells. These type of lymphocytes, they're called T because they mature in the thymus, which is kind of in the neck. There's two subcategories. There are what we call the T helper and the T cytotoxic. Sometimes the T helper cells are called CD4+. CD stands for cell differentiation. So these have a receptor on them called CD4, which is sitting on the receptor of the outside of the cell, of the lymphocyte. Whereas the cytotoxic T cells, these are CD8, meaning they have a different receptor to these guys, being 8, which is on the outside of these cells. Also, like we saw over here, at this point in time before they are primed, they are what we call naive. So how do we get the selection for and the priming of the adaptive immune system to respond to a specific infection? Down here I've drawn a bacteria, which is a green particular cell with these red flags over it. The red flags are there to indicate an antigen, which means something that is foreign that your immune system will react to specifically. Remember, specific means adaptive. So this bacteria, let's say, has breached and gone into your finger. It's overcome the two barriers of your innate immune system, being your skin and so forth. So it got through, there's a bit of inflammation, etc., but the bacteria are replicating. So the bacteria is starting to cause problem. So we need to bring in more arsenal to come in and specifically get rid of it. Okay? So with the bacteria will infect your cells in your finger. Now there'll be some cells that will get infected directly by it. So some cells, let's say, will get the bacteria incorporated into it. These could be phagocytes, or these could just be any type of cell in your finger that could be infected. Now, it's less likely bacteria to specifically invade the inside of your cell because they're more extracellular. Viruses are more intracellular, but let's just say for this case, this bacteria has gone in, infected one of your cells in your finger, and now it's damaged, dysfunctional, and possibly may die. Before it does so, there are receptors on the outside of your cell. Okay, all your cells have this. And in this particular case, this one is called the MHC1, meaning major histocompatibility complex 1 receptor. So this will present a type of, let's say, antigen on it, okay, which will present on the outside. It's not an antigen presenting cell, but it's just a part of the antigen with the bacteria that's presenting on this MHC1 receptor. Now, this will present to, or this will come into contact with the CD8, the cytotoxic T cell. Okay, through a communication between these receptors, what it will do is it will find the right type of CD8 cell that has a receptor that will fit into this receptor with this type of antigen. So there are thousands and thousands of different type of naive CD8 cells, but there's a particular one that will lock into this type of cell with this type of antigen, and now it's activated. So now this cell is activated, this type of CD8 cell, which means that this cell is now selected for and now primed. Okay, what it will do is it will go through clonal expansion. So it will clone itself. Clonal. clonal expansion, which means it will clone itself, just keep copying that type of CD8 cell that is specific to that type of antigen. Now there'll be two groups of cells that it will make. I'll get a blue one. One group will be a memory CD8 cell, and one will be a cytotoxic T, which just means that's the one that's going to do the killing. This one will go kind of down and want to start doing more killing, but we'll leave it at that point. I just want you to know, once it's selected for, it will go through a level of clonal expansion, which is going to make two subgroups, a group of memory cells, which are just going to hang around in case they encounter this bacteria again in weeks to months, or a group that's going to go and try and kill cells that have been infected with the bacteria. So that's one part of it. There's going to be also some cells that will phagocytose it, so it will actually eat them up. So there'll be some cells that will eat it and swallow it, and you'll have the bacteria inside it, like with the antigen. So this cell will start to gobble, chew it up, and what it will do is it will then, through its own receptor on the outside, present it. So it will put the red flag on the outside to say, hey, we've got an antigen for a bacteria that we want to show you. This particular, and this is what we call an antigen presenting cell, and these can be dendritic. Dendritic cells are one of the most common. They are found in the skin, or it could be also macrophages and other antigen presenting cells. Even B cells can do it, but we'll just stick with a dendritic cell. This will come and bind to a CD4 helper cell, which is also naive at this point. Again, like we saw here, there are thousands of different subtypes of naive CD4 cells, but there will be one that's particular for the one thing I did forget to mention. This type of receptor is a MHC2, unlike this one over here, which is MHC1. So this one, with the antigen, will select for a particular type of CD4, which then activates this one, just like we saw over here. Now it is primed, which does two things. One will become a memory. One will become memory CD4, just like we saw the memory CD4, just for CD8, like we saw last time. This is for a later date in case we encounter the bacteria again. But another group will just go through and copy itself through cloning itself again. So clonal expansion, which means we make heaps of CD4 plus for that particular antigen. And what do these cells start to make? Lots of cytokines. And what do these cytokines do? Well, they'll notify your other branches of your immune system to come along and do some more killing. So some examples are natural killer cells, some macrophages, but also B cells. So these are chemical cytokines that flooding the finger full of cytokines like interleukins, etc., which will bring more natural killer cells, more macrophages along, which with the communication of this particular type of receptor antigen, those two types of cells can come and select for the bacteria and just start killing all that bacteria. But the B cells, what it will do is it will send a chemical signal across to here to tell the B cells, at this point, they're naive, to select for the B cell that is specific for this antigen. And again, there are thousands and millions of different types of B cells. On the surface of the B cells, we have this thing called an antibody, which is that black Y thing I've drawn there. So there's going to be one particular type of B cell that is specific to this red antigen, and when it selects for, through a communication here, it will activate the B cell. So now it becomes primed. Now what the B cell will do is two things. It will also clone itself. One group will become memory B cells, and the other one will become plasma cells. Now, what are the plasma cells? They are basically just filled with antibodies. So they are just filled with antibodies. And these antibodies are selected for or specific to that type of antibody from that B cell, which was selected for from this particular CD4 cell, which was selected for from that bacteria's antigen. So that means all of these antibodies are now just specific only to that antigen. And they start to get released into blood. And this is essentially what the humoral part is. So now your blood's filled with these antibodies, particular just for that one antigen. So once it floods through your body, if there's bacteria else in the body, the antibody will bind to it. So the antibody will bind like this, like that. Now this will do a few things. When it binds to it, a flash will go off like a siren, which will bring on, say, the cytotoxic T cells, which will go and kill them, and also bring in the other macrophages and the other cells that can do the killing and kill those. But also the other antibodies, what they will do is they will stick together. So all these antibodies will cluster in together, and the bacteria will kind of cluster in and come out of solution, which will also tell the macrophages and the neutrophils to come along and get rid of it. They will also neutralize it. So if the bacteria has certain harmful chemicals or toxins, it will neutralize it to stop any more injury. It will also activate the inflammatory response. So more cytokines will be released, more histamine, so more inflammation, more white blood cells will come to the site. It will also activate the complement system. So the complement system, which we saw in the innate system, will come along and bore holes into it and kill it. And opsonize it, which means, again, it kind of clusters it up, brings it out of solution, and can be killed off. So that is essentially the specific or the adaptive response to a particular bacteria. Now, once this bacteria has been killed, there's no more bacteria in it. Because we have memory cells, memory, that means when the bacteria comes along again with the same antigen, maybe in a month's time or a year's time, your immune system is much more efficient, and it's a lot quicker because it has memory to it. Okay, it doesn't have to go through all this clonal expansion, which takes days to maybe even weeks to get to the point of overcoming this bacteria. It's much quicker, and it can get rid of it much more efficiently. And this is the basis of where we do vaccines or immunizations, is you get memory to it. So when you actually encounter the real infection, you're very efficient and you can get rid of it. So hopefully what you've seen is this particular system is specific. The case that we gave was this particular bacteria, which has a type of antigen which activates the naive T and B cells, which can do a combination of cytotoxic, which is killing the infected cells directly, or helping the CD4 cells, helping prime the other parts of the immune system, particularly the phagocytes and the B cells. And the B cells, primarily using antibodies, can also help with the way that the antibodies bind to the bacteria or the infection, and also bring other recruitments in to get rid of the infection. So hopefully what you've seen for this video is what the immune system is, the different components, the two arms being the innate and the adaptive, and how it all works together to fight infection.